Welcome to the Viral Blueprint, Form, Function, and Taxonomy. In this presentation, we will explore a structural and operational framework for classifying obligate intracellular parasites, commonly known as viruses. My name is Triwibowo Uono from the Department of Agricultural Microbiology at Universitas Gadjah Mada, Yogyakarta. To truly understand viruses, we must first grasp the concept of the obligate intracellular paradigm. Unlike cellular life, which exhibits growth, division, and self-sustaining metabolism, viral entities are fundamentally different. Cellular life typically possesses a double-stranded DNA genome exclusively. In contrast, viruses engage in de novo replication, often utilizing mobile genetic elements and possessing either DNA or RNA genomes. A defining characteristic is their metabolic inertness outside a host, meaning they require a host cell to replicate and produce at least one major virion protein. The scale of viral architecture is truly microscopic. To put it into perspective, an average human cell is about 10 to 30 micrometers, and a typical bacterium is 2 to 3 micrometers. Viruses, however, are significantly smaller. For instance, the Pandora virus measures around 1,000 nanometers, while megaviruses and poxviruses are about 400 nanometers. Smaller still are the influenza virus at 80 to 120 nanometers, HIV at 100 to 130 nanometers, and the tiny picornaviruses, which are only 20 to 30 nanometers in size. The classification of viruses presents a unique biological divide compared to cellular life. Traditional Linnaean taxonomy for cellular organisms follows a hierarchy from domain to species, based on common ancestry, reproductive units, and ribosomal RNA lineage. Viral taxonomy, governed by the International Committee on Taxonomy of Viruses, or ICTV, also uses a hierarchical system, but its core basis relies on polyphyletic class criteria. These include molecular makeup, capsid structure, host range, and genetic sequence similarity. Crucially, viruses do not fit into the cellular kingdoms, necessitating their own distinct classification system. Viruses exhibit diverse architectural boundaries, primarily categorized into naked and enveloped virions. A naked virion consists solely of nucleic acid and a protein capsid. Its attachment to host cells relies on these capsid surface proteins. Examples include adenoviruses and picornaviruses. In contrast, an enveloped virion possesses an additional lipid bilayer, derived from the host cell, along with matrix proteins and glycoproteins. These viruses use attachment proteins embedded in their envelope to bind to host cells. Well-known examples are HIV, influenza, and coronaviruses. The limited genomic size of viruses means they cannot encode large, complex shells. Instead, viral capsids rely on the self-assembly of repeating protein subunits called capsomeres. There are three main types of capsid symmetry. Icosahedral capsids are spherical or prolate, offering exceptional stability, as seen in adenoviruses. Helical capsids are rod-like or filamentous, where the nucleic acid defines the structural length, exemplified by the tobacco mosaic virus and Ebola. Finally, complex capsids do not conform strictly to helical or icosahedral forms, such as those found in poxviruses and bacteriophages. The genomic matrix of viruses is broadly divided into DNA and RNA viruses, each with distinct characteristics. DNA viruses can have double-stranded or single-stranded genomes, which can be linear or circular. They are generally highly stable due to the presence of proofreading DNA polymerases. Examples include herpes viruses, pox viruses, and parvoviruses. RNA viruses, on the other hand, can also be single-stranded or double-stranded, linear or segmented, and can be positive or negative sense. They are characterized by high mutation rates, often 10 to the power of -4, driven by error-prone RNA replication, leading to rapid adaptability and variant generation. RNA viruses account for approximately 70% of all classified viruses, with notable examples like SARS-CoV, hepatitis C, rubella, and HIV. At the heart of viral replication lies the Baltimore engine, which describes the pathway to positive sense messenger RNA, or plus mRNA. This is a fundamental concept. Regardless of its genome type, every virus must synthesize plus mRNA. This plus mRNA is then used to hijack host ribosomes, allowing the virus to translate its viral proteins and replicate. The Baltimore classification system categorizes viruses into 7 distinct classes based on their genome type and mRNA synthesis pathway. Class 1 includes dsDNA viruses like adenoviruses and pox viruses. Class 2 comprises plus ssDNA viruses such as parvoviruses, which first convert to dsDNA before producing mRNA. Class 3 consists of dsRNA viruses like reoviruses. Class 4 are plus ssRNA viruses, including coronaviruses and picornaviruses, which directly use their genome as mRNA or produce a negative sense RNA intermediate. Class 5 are minus ssRNA viruses, such as orthomyxoviruses and rhabdoviruses, which synthesize mRNA from their negative sense genome. Class 6 are plus ssRNA RT viruses, like retroviruses and HIV, which use reverse transcriptase to produce a DNA intermediate. Finally, Class 7 includes dsDNA RT viruses, such as hepadnaviruses, which also use reverse transcriptase. The ICTV taxonomic funnel provides a structured approach to viral classification. At the highest level, order is determined by general biochemical composition, replication strategy, and overarching genome organization. Moving down, family classification considers the nature of the particle structure and specific genome organization. Genus is defined by genome size, the number of segments, sequence homologies, and vector transmission. Finally, species are monophyletic groups characterized by high sequence homology, antigenicity, host range, tissue tropism, and pathogenesis. To ensure consistency in viral nomenclature, specific syntax rules are followed. The suffix directory provides standardized endings for each taxonomic level: Viria for realm, Viricoda for phylum, Virichetes for class, Virales for order, Viridae for family, Virinae for subfamily, and Virus for genus. Orthography rules dictate that formal taxonames, such as order, family, genus, and species, must be written in italics with the first letter capitalized. Vernacular or common usages, however, remain in lowercase Roman script. Importantly, virus species names are designed to be distinct and must not merely contain the term virus and the host name. Let's synthesize our understanding by examining the complete viral profile of human immunodeficiency virus 1, or HIV-1. Physically and operationally, HIV-1 is an enveloped virion with a conical or icosahedral capsid core containing 2 copies of linear positive sense single-stranded RNA. According to the Baltimore engine, it belongs to class 6, meaning it's a plus ssRNA RT virus that uses reverse transcriptase to build a DNA intermediate before producing mRNA. Its taxonomic lineage places it in the realm Riboviria, kingdom Pararnavirae, order Ortovirales, family Retroviridae, genus Lentivirus, and species human immunodeficiency virus 1. The family name Retroviridae directly reflects its reverse transcriptase capability. In summary, decoding the virosphere involves understanding the intricate science of virus classification. We've covered the structural and physical foundations, including enveloped versus naked virions, capsid symmetry and shape, and host-based categorization. We also explored the genomic frameworks and formal taxonomy, delving into the Baltimore classification system, the ICTV hierarchy, and the standards for formal nomenclature. Thank you for joining this exploration into the viral blueprint. I hope this session has provided a clear understanding of viral form, function, and taxonomy. I look forward to seeing you in the next class.